whose land we're meeting upon today, and whose land I have done my work on, as well as acknowledge the fact that it is Mana Whenua has protected the Ngāhere for generations and will continue to protect the Ngāhere going forward, and hopefully in concert with us and the information we're providing. So with that, I'm working on the conservation of maire tawake, or wairaka, or swamp maire. So depending where you're from, you'll know the different names. It's a taonga species for many people I've spoken to, and many groups. And it is also a myrtle, or a myrtaceous, so it's affected by myrtle rust. So with that, I thought I'd just start here. What are the current tools that we have against myrtle rust? So we've spoken about many things that are sort of being developed, and that really all that we have right now is removing infected material or spraying with fungicides, and both of these are extremely damaging to the environment, extremely costly. And the other thing that we can do is breed for resistance, but as Grant just spoke about, we don't really understand resistance, and these are big, large-scale trials that we need to do. So instead of that, we can start looking at the genetic diversity of species and conserving that. So within populations, we already have much variation, which can allow certain individuals to be resistant to pathogens or other things such as climate change. So as Stacey demonstrated earlier when she asked who in the room had COVID, there are certain individuals who are naturally more resistant to diseases, and this is the case for trees and animals and all other organisms. So with that, I just wanted to introduce something about maire tawake. So its scientific name, Western name is Syzygia maire. But as I said, it already has many other names. It's a major component of North Island swamp forest. You can see the distribution over here on the right. That was mapped by Manaki Whenua a few years ago. It also has fruit that's really important for birds, and many uses to Māori as well. Blake pointed out earlier that it's known as weaponwood. It also, the berries were edible, and it's been used for treating ringworm, as far as I'm aware. But regardless of that, it's currently classified as nationally critically threatened. And the two major reasons for this are, number one, habitat loss. So it grows in lowland areas, which are swampland, which has been drained mostly for farmland, but also cities and towns. So it's lost a huge amount of habitat. And then, as I said, myrtle rust. So it was alluded to by Michael Bartlett earlier, but maire tawake seems to be just as susceptible, if not more, as ranganga to myrtle rust, with infection on young tissues, but also flowers and fruit, meaning that although we have trees standing in the landscape, they might be undergoing functional extinction, which means that they just can't breed, they can't produce offspring, and future generations are not able to persist. A big problem here is that we also haven't figured out a way to preserve seed, and it doesn't persist in the seed bank. So whatever seed we have one season, that's all there is. So if that tree dies that season, we're not going to be able to preserve any of its genetics or going forward. Another major point that I wanted to make, and I promise this is the only graph in my presentation, and I'll have pictures, is that restoration doesn't ensure adaptability. So we really need to ensure that we're planting out the full genetic diversity, and that's because during habitat loss or eco-sourcing, and I just want to here say that I mean eco-sourcing in the sense of trying to preserve the local genetics of the species, not necessarily speaking about mataranga or whakapapa of that species, because that's a different conversation. I've heard that used as cultural sourcing as opposed to eco-sourcing. But here, when we lose habitat or restore plants, especially when we're sourcing seeds just from easily to access trees or just single mother trees, we have a huge loss in genetic diversity. Now, even if population size increases after that, we don't see a corresponding increase in genetic diversity, and that's because that genetic diversity has been lost from that population. So what we have now, we really need to work on preserving. But the only way to preserve that in an informed manner is to understand what's there already. So that's where my project comes in. So before you can assess the genetics of many species, at least in a holistic manner, you need to have a reference genome. So that's understanding how all the genes in a genome are arranged, which chromosomes they map to. So for instance, if we wanted to look for resistance to myrtle rust or drought or something like that, we would understand where genes might be on the genome and where to look for resistance. So Kaurilands or Manawatu has is acting as kaitiaki for a genome which we've sequenced from one tree within the rohe at a site called Eco Viaduct. They are currently undergoing a naming process for this genome and will hopefully be acting as co-authors in any papers that we put forward, ensuring that the mataranga and of this tree is progressed forward and understood amongst everyone. So we've constructed this genome. So this is a big messy figure, but what it's showing is on the left we have the maire tawake genome or swamp maire genome, and on the right we have clove, which is another syzygian species, which is quite closely related and has a really highly complete genome also assembled. So this is just comparing one by one each chromosome to a chromosome on the clove genome, and what we can see is that we've got a really complete genome. We've been able to assemble and sequence everything that's in maire tawake as compared to clove. So 10 years ago, this would have been largely impossible. It would have been extremely expensive, and we would have just got thousands of little chunks. But with new technologies, it's becoming a lot cheaper, and I did this within a year, and we could possibly continue to do this for many of Aotearoa's other taonga species within the scope of smaller projects such as Ngā Rākau Taketake. The second question that I wanted to ask, and I'm just starting to work through much of this data, was how diverse are trees across Aotearoa? So I've collected 301 adult trees from across the Motu, from across the North Island, and one little population on the South Island, which is the only one that's known there, and we don't know how much more there may have been there. So this is in order to assess the remaining diversity of the species as well as the distribution of diversity. So this could be akin to whakapapa in purely the genetic sense, and eventually the effects of fragmentation. So how much diversity might there have been that we expect to have been lost after habitat loss, and how can we ensure that isolated populations are able to still breed together and continue that diversity. So I collected trees from up in Waipoa. So if I say Northland from now on, I mean Waipoa. I didn't collect from elsewhere in Northland, unfortunately. Bay of Plenty, Taranaki, and down at the South Island. Yeah, I didn't collect from Auckland and around the Coromandel, unfortunately. I was doing this in 2020, 2021 during COVID, and it just was not possible. I would love to include that in the data, but yeah. So just in really quickly, because I got the yellow card, this is a statistical analysis which groups individual trees, which is one of these little dots here, into clusters based on how genetically similar they are. And what we're seeing is about 5 main groupings across the North Island, 4 main groupings across the North Island, where we're getting the Manawatu and Greater Wellington populations grouping together, as well as populations outside of, and the rest of the populations grouping separately, and the South Island population looking quite different. I also looked at the relatedness of trees across the North Island, and at the top is Northland, and as we go down, we're looking south. So the diagonal from the top left corner to the top bottom right corner is inbreeding. So if it's really red, the trees are really inbred, and everything else is comparing each tree to every other tree that I sampled. So this is everything in Taranaki compared to all the other populations. And as we see higher or more red colors, the trees are more related, and we're seeing increased relatedness as we move southward, suggesting that there may have been an ancestral population up in Northland, and trees migrated southward. Thank you. So that brings me to my conclusions, which is that genomes for Aotearoa species enable us to answer conservation-focused questions. We might see 4 to 5 genetically distinct populations in maire tawake. And really the take-home for everyone here is that restoration projects could consider broader eco-sourcing zones, informed at a more local scale by whakapapa, paturanga, or tikanga. And I just want to say thank you. And my email is down there, but please approach me if you have any questions afterwards. Thank you.
Kia ora.